flagella. Today we will learn about gram staining procedure. The steps involved in gram staining. Based on the gram staining procedure, we are classifying bacteria as gram positive and gram negative bacteria. Okay, so then we will learn about respiration in bacteria, nutrition in bacteria and reproduction in bacteria. So these are the topics we are going to learn today. So whenever you listen to the lesson, please have your classwork with you and you have your book beside you so that you can refer to that. Then whatever I teach, you observe, please put it in the classwork then and there. When you listen, you please write in your classwork and you can submit it. Yes, so we will start with bacteria gram staining procedure now. So gram staining procedure involves series of steps and why we have to stain the material. The bacteria should be stained for clear observation. Whatever be the material, you should have observed a slide. Slide is in pink color or it will be in violet color generally. So that is stained for clear observation under microscope. When a material is not stained, you will not be able to see the material clearly. Okay, that is the main purpose of staining the material. While staining the material, the bacteria was observed to be violet in color and some of the bacteria are appearing only red in color or pink in color, which means they do not have the capacity to hold the stain. So based on this characteristic feature, they are classified as gram positive and gram negative bacteria. So we will see the steps involved in gram staining. First we have to make smear. What is a smear? It is a thin layer of material for observation. So you have to take a material and you have to make it very thin on top of the glass slide for observation. Why should it be made thin? If it is not thin, you will not be able to view the object clearly because light has to travel through the object for you to view the object clearly. Follow? If the light travels through the object only in the microscope, you will be able to see the object clearly. So it should be thin. How to make the smear? We are going to make a bacterial smear. For that we have to take the bacteria from the culture medium. Consider this as a culture vessel. It is a glass plate where you have the medium made of agar-agar and other nutrients. There you have the bacteria growing. You have to take it, put it on top of the slide and make it thin. Just make it very thin. Then you have to move on for the next step, adding crystal violet stain and leave it as such for 30 seconds for it to absorb the color. Then you have to rinse it with water for 2 seconds. After that, you can use grams iodine stain. It is also in the liquid form. You have to put it over the slide and leave it for 1 minute. After 1 minute, you can rinse it with distilled water. After rinsing that, you can use the ethanol. And then you have to put the ethanol or acetone over the glass slide. Then you have to use distilled water so that all the excess water in the slide will be removed. Then you can use saffron, that is a stain which is looking in red or pink in color, for 30 to 60 seconds so that the bacteria will also absorb this color. Then you have to use distilled water to clear extra stains. Okay? After that, you can blot it. So what is blot? There is a paper, it is like tissue paper, that is called blotting paper. That if you put it onto the slide, it will absorb whatever is excess material left over in the liquid form. Okay, so blotting paper is used to remove the excess water or excess liquid material over the slide. Then you can observe it under microscope. Okay, when it was observed under the microscope, some of the bacteria were appearing in violet color. Some of the bacteria were appearing in pink or red in color. Pink or red in color. These bacteria are classified as gram-positive and gram-negative bacteria. We are using gram staining procedure. When it is responding, it is taking up the color. Then it is called as positive bacteria. When it is not taking up the violet color, then it is called as, it is not responding to gram staining. So it is called as negative bacteria. So gram-positive bacteria and gram-negative bacteria stains differently. That is one difference. And they have different chemical and structural compositions. Okay. So when you see the chemical composition, gram-positive bacteria shows the presence of teichoic acid, teiuronic acid and polysaccharides, which you don't see in the case of gram-negative bacteria. Then they have a rough surface. They have the surface rough. So it is able to hold the stain and appear violet in color. Understood? So here in negative bacteria, presence of lipids, lipoprotein, lipopolysaccharide is more, and it gives elastic nature to the bacterial cell wall. This elastic nature does not hold the stain, the violet stain. It is not able to hold. It just leaves it outside because of the presence of this lipids, lipoprotein, lipopolysaccharides. So that it is just leaving it as such, so it appears pink or red in color. Then what are the other differences? It is highly susceptible to antibiotic. Gram-positive bacteria are highly susceptible. They are highly prone to. They are easily affected by penicillin. Then low susceptibility is seen in the case of gram-negative bacteria. Then the basal body rings. You should understand the term basal body rings to understand the difference between positive and negative bacteria. Basal body is nothing but the structure from where the flagella arise. You know from where the flagella arise. So it is not from the outermost layer of cell wall. It is just inner to the cell wall, that is plasma membrane. So in the plasma membrane, from one particular point only this flagella arises. That is what is called as basal body rings. Okay. So if you can understand all these terms, you can learn the difference between the gram-positive and gram-negative bacteria. Next we shall see about respiration in bacteria. What is respiration? Tell me, what is respiration? Generally, we say it as taking in oxygen and leaving out carbon dioxide. The general process of taking in oxygen, leaving out carbon dioxide is general breathing. Respiration involves the actual process of utilizing oxygen for the release of energy in the form of ATP molecule. Yes, so you should know respiration does not only deal with this breathing process, it deals with the utilizing oxygen to produce energy in the form of ATP, and it occurs in cellular level. It occurs at the cellular level. Okay, so bacteria being a unicellular organism, it shows these types of respiration. Okay, so what are the types it shows? Aerobic respiration. What is aerobic respiration? When it utilizes only oxygen for its survival. It is able to take only oxygen and it is They are called as aerobic bacteria. Under aerobic bacteria, we have another type called as obligate aerobic bacteria. They can definitely not be living when there is no supply of oxygen. They are strictly aerobic bacteria. They need the presence of oxygen for its living. So that is about aerobic and obligatory aerobic bacteria. Then anaerobic bacteria. Anaerobic bacteria can live in the absence of oxygen. That is called as anaerobic bacteria. Then we have an interesting kind of bacteria that is called as facultative bacteria. So these facultative bacteria, they can withstand the condition where there is no supply of oxygen, and it can utilize the oxygen when there is a supply of oxygen. Understood? I'll tell you the example now. Bacteria E. coli, which is able to live in the presence of oxygen, it absorbs all the oxygen for its survival. Once it is absorbing the oxygen, it makes the condition without oxygen. It makes the condition anaerobic. E. coli is able to make the condition anaerobic or no oxygen condition. Some of the bacteria can live only in the place where there is no oxygen. Example is Salmonella. Salmonella is a bacteria. Have you heard of the bacteria Salmonella? Salmonella is a bacteria which is able to live only in the absence of oxygen, and it causes the disease typhoid. Salmonella typhimurium. Okay, so typhoid is a bacterial disease. Which part does it affect? When a person is suffering from typhoid, which part is getting affected? Do you know? What does the doctor suggest? They suggest strict diet, right? So it affects the digestive system of a person. Okay, so this E. coli, it is making the place oxygen free, and it allows the anaerobic bacteria Salmonella typhimurium to grow in the intestine and causes abscesses. The word as it is given in your book is abscesses. Abscesses is like a little tumor like. Okay, it just makes the ulcer, the next condition to ulcer. Okay, so this abscesses is made by this Salmonella. They are called as facultative anaerobic bacteria. Okay, so the next condition is canophilic bacteria. So it is very interesting. Canophilic bacteria can survive in the presence of carbon dioxide. They like to live in the presence of carbon dioxide. They are called as canophilic bacteria. So that is all for respiration in bacteria. Understood? What are the types of respiration in bacteria? Can you recall? Aerobic, anaerobic, then facultative, then canophilic. Okay? So next we shall move in for nutrition in bacteria. What is nutrition? The mode of feeding and utilizing the food is called as the nutrition. Okay? So the nutrition in bacteria, first we shall see about autotrophic. What is autotrophic? So usually auto means self, trophic with reference to the mode of nutrition. Okay, mode of feeding. Auto means self, it is able to synthesize its own food. Where do you come across this term generally? In the case of plants, they are autotrophic. So whenever you see a word, you should remember the term light. It refers to light. Photoautotrophic bacteria utilizes light and it can synthesize its own food. Okay. So here the other type of bacteria which cannot utilize light, the body system is not such that it can utilize light. So they are called as chemotropic bacteria. Chemotropic bacteria. Understood? So phototropic and chemotropic bacteria. So under phototrophic bacteria, we have two types. Under phototrophic bacteria, we have two types. They are photolithotrophs and photoorganotrophs. Photolithotrophs and photoorganotrophs. Here, chemotrophic also, they are lithotrophs and organotrophs. Okay, so the photolithotrophic bacteria means they have to use the substrate which is not organic in nature. Okay, inorganic material is used as a substrate. In the presence of light, bacteria is able to synthesize the food. They are called as autotrophic, photoautotrophic, photolithotrophic bacteria. Based on that, the photolithotrophic bacteria, two types of bacteria you are going to study in your lesson is green sulfur bacteria and purple sulfur bacteria. So they have a special material for this purpose, that is bacterioviridin and bacteriochlorophyll. Bacterioviridin is present in green sulfur bacteria. Bacteriochlorophyll is present in purple sulfur bacteria. They are using the substrate as hydrogen sulfide in the case of green sulfur bacteria. Thiosulfide is used in the case of the purple sulfur bacteria. Understood? Next, photoorganotrophic bacteria. So what is organic material which has got the carbon compound? So this photoorganotrophic bacteria will utilize alcohol as a substrate and it is able to synthesize its own food. Understood? So photoautotrophic bacteria is over. Next we will go on to chemoautotrophic bacteria. So under chemoautotrophic bacteria you have lithotrophic bacteria. What is lithotrophic? Litho means inorganic material is used as substrate. This bacteria can use iron, sulfur, hydrogen as the substrate material and it is able to synthesize its own food, that is chemolithotrophic. Okay, then coming to chemoorganotrophic bacteria. So here the organic material is used as a substrate. What is the substrate used? Is the methane, acetic acid and lactic acid is used as a substrate for the production of energy. That is called as chemoorganotrophic bacteria. Understood? So next we shall see about the other type of nutrition. So generally how can we describe the nutrition? Either it can be autotrophic or it can be heterotrophic. So far we have seen about the autotroph. Next we will see about heterotrophic nutrition in bacteria. Bacteria can be parasitic or it can be saprophytic. Okay, so what is a parasite? What is the difference between parasitic and saprophytic heterotrophic bacteria? Parasitic bacteria lives inside the living body. It requires the presence of living cell to absorb the nourishment. But saprophytic bacteria lives on dead and decaying matter. That is what is called as saprophytic bacteria. Next we shall see about reproduction in bacteria. What is the reproduction? Last class I told you. What is the reproduction? Again. Production, producing. Producing of its own kind. That is called as a reproduction. Or it is also called as bacterial genetics. So reproduction in bacteria we will learn under conjugation, okay, transformation, and transduction. These are the three methods that we are going to learn under a reproduction in bacteria. That is conjugation. Here two bacterial cells are involved. Okay, one is recipient. Recipient means receiving. The other cell will be the donor cell. That is about conjugation. It is a kind of sexual reproduction. Transformation. The uptake of bacterial genome through the cell wall as such. There is no tube, there is no connection. Simply the transformation occurs. The genetic material just enters into the bacterial cell. That is what is called as transformation. Okay. Next, transduction. Here the genetic material is transmitted. It is transformed from one to another with the help of virus. That is bacteriophage virus. Okay. So conjugation tube is involved. Transformation, no tube. The transformation of genetic material occurs through the cell wall. Transduction, it requires the help of virus for moving the genetic material from one bacteria to another bacteria. Okay, we shall learn conjugation in detail now. About conjugation in bacteria. So, two bacterial cells are involved. Two bacterial cells are involved. One is acting as donor, other one is acting as a recipient. Donor cell and a recipient cell. This is denoted as a plus and this is denoted as a minus. Okay, so we learned about the Fimbriae when we studied about ultrastructural bacteria. It is a special type of flagella which helps in the conjugation process. Okay, so this is getting attached with the recipient cell to form the conjugation tube. Okay, so for the conjugation to occur, we need the presence of two bacterial cells, donor and recipient, and the formation of conjugation tube occurs with the help of Fimbriae that is occurring in the donor cell. Okay, so now for the reproduction or for the sexual reproduction process, there should be a movement of genetic material should occur. Okay, so usually we have the genetic material that is called as incipient nucleus or nucleoid, and another one is called as the extranuclear material, extra chromosomal material that is called as plasmid. During the process of conjugation, this plasmid is cut and a layer is moving on to the recipient cell. Okay, this plasmid is moving on and it is coming into the other cell. When we studied about plasmid, I told you they are self-replicating in nature and they are autonomic structures. Okay, so they will replicate another copy of it and forming another layer, and here the same thing happens to form a full plasmid. Okay, so once the genetic material transformation gets over, these bacterial cells segregate themselves from each other. Okay, so during this process, what happened now? This recipient cell has received some genetic material, and whichever is being received, that will be expressed by this cell. Before, those characters will not be expressed by them. Now, after receiving the plasmid, the genetic material which is present in the plasmid will start bacterial cell is made. This bacteria will undergo the normal process of asexual reproduction, that is binary fission. So increase in number of copies of the bacterial cell will be produced. That is how the reproduction occurs and different combination of bacteria is produced. So that is for conjugation in bacteria. The next method, do you remember what is the method? Yes, correct. Transformation. Transformation is a method in bacteria that was proved by the scientist Griffith, and it is explained by the Griffith experiment using mouse. So we will learn about Griffith experiment in detail. So materials used are the mouse and Diplococcus pneumoniae is a bacteria. This Diplococcus pneumonia shows two types. Diplococcus pneumoniae bacteria are of two types. One is a rough, other one is smooth. Okay, so based on the structural outlook, how it looks, okay, the smooth bacteria and the rough bacteria. And you have to understand the term virulent. What is virulent? Virulent is which will cause adverse effect. That is called as virulent. They are very active. Okay, avirulent. It is not so active as virulent bacteria. That is what is meant by avirulent. Okay, so the smooth type of bacteria is found to be virulent. It is causing severe adverse effect in the organism. So smooth is of virulent type and rough is of avirulent type. What are we talking about? The bacteria Diplococcus pneumoniae. Okay, so this Diplococcus pneumoniae bacteria, avirulent strain, that is a rough strain. Strain means what? It is a type. Strain means this type. R-type bacteria is injected into the mouse. Nothing happened to the mouse. It lived its life without any defects. Virulent strain, yes, was injected into the mouse and the mouse died. Because of its virulence, it affects the body and it is making the organism die. Then he wanted to test with the heat-killed S type. Once this virulent type is killed by heating process. Okay, so killed by heating process, the outer coating is getting heated up and it becomes inactive. Once it is heat killed and it is injected into the mouse, mouse does not show any change, it lived happily. But then he wanted to try with the combination of heat killed smooth type, that is virulent, and plus avirulent rough type. When he just mixed both rough and smooth heat killed and it was injected into the mouse and the mouse died. So when we wanted to find the reason for the mouse to die when it was injected along with the rough strain, he found that the method of transformation occurred from S type to R type. Okay, so smooth type has got the genetic material that has nothing has happened to that when it was heat killed, only outer coating got affected. Okay, so that material is moved on to the rough materials. If I consider this as a bacteria, that is S type, consider this as a bacteria, that is R type. The genetic material from S type has just come out and it is just moved inside through the cell wall into the rough type. So this rough type becomes virulent and it started killing the mouse. So this is what is the experiment done by Griffith to prove transformation occurring in bacteria. What is the next type of reproduction I told you? What is that? Good. Transduction. So what is transduction? What is required for transduction? Transduction involves the presence of virus. Virus helps in the transmission of the genetic material. So you have a bacterial cell. So bacteria is getting affected by virus. So once it is getting formed, it has to get enclosed with the genetic material. During this process of encasing itself with the genetic material, by chance the bacterial genome gets inside. Bacterial genome or bacterial gene goes inside. Understood? Actually, viral genome only should go inside. Accidentally, it can carry the bacterial genome. Now we are bothered about only this virus. So you follow this virus now. This virus is having the viral structure outer, okay, and whichever material is being present inside is a bacterial gene, bacterial genome. Okay, what is its role? It will automatically go and affect the bacterial cell. So another bacteria is getting affected by this virus. What will happen? It will automatically inject the bacterial gene. Okay, then whatever genetic material is there in that bacteria will get infused with this bacterial gene. Now the fusion of two different bacterial genetic material occurred by transduction, that is with the help of virus. This is what is another kind of reproduction seen in bacterial cell. Understood? So far we have seen about the sexual reproduction in bacteria. Now we will see the asexual reproduction in bacteria by binary fission. What is fission? Fission means separation. Okay, so once a bacteria grows to the maximum size, it is having enough cytoplasm and the nuclear material. Okay, so it starts dividing, the cleavage occurs. First, the cleavage of cytoplasm occurs. Okay, so movement of the cytoplasm occurs. Then the nuclear material move to the corner. Okay, two corners. So then they will get detached. Once it is becoming thin in the middle, it will just get cut. Okay, so once it is just moving to the corner, it will just make another copy by developing the cell membrane and cell wall. Then it will grow to its maximum size and it will increase in number. Understood? So one bacteria has become two. It has reproduced. One more bacteria is produced. Okay? By the process of elongation of cytoplasm and the genetic material. Do you remember where we studied about this? During the mesosome. Mesosomes are the folded part of the plasma membrane. During this process of binary fission, they just extend itself to form another cell. Remember, refer to that video also. Okay, so this is called as binary fission. Next we have the condition of endospore formation. So what is endospore formation? During unfavorable condition, this bacteria is becoming rounded off in its structure. Bacterial cell will be like this. Okay, so this bacteria is getting rounded up and it forms encapsule. Encapsule is the thickened coating around it. Okay, so this is called as encapsulation and it becomes inactive. Inactive bacteria. It stays as such during unfavorable condition. Once the favorable condition returns, it starts multiplying on its own by the method of binary fission. So example for this is the Clostridium bacteria that is found in the soil. Have you come across the word TT injection? Okay, so why we are acting against this Clostridium tetani. Clostridium tetani is a bacteria that is found in the soil. If a person happened to fall down to the soil, if he is getting the bacteria by chance into his body, that will be the favorable condition for the bacteria. Then it will start growing and it will cause a disease called tetanus. So why I am saying is, it is the endospore of Clostridium is found in the soil in the inactive form, that is endospore. During the condition of favorable condition, it will start multiplying itself. Okay, so we have studied about the reproduction in bacteria. What are they? Asexual reproduction during favorable condition, binary fission, unfavorable condition, endosperm formation, sexual reproduction, conjugation, transformation, under transformation, Griffith experiment, and transduction, how it occurs with the help of viruses. Okay, next topic is economic importance. Under that you have how the bacteria is used in soil fertility and how it is used in the production of antibiotics against the bacteria is used. Okay, so that is the antibiotics and the industrial uses. When we want the product in a large quantity, we go for industrial production. In that field also the bacteria are used. Please go through the economic importance, beneficial effects of the bacteria given in your book. Okay, now it's time for homework. You have to take down the notes. When I took the lesson, I told you to copy as a notes. That should be there in your classwork. Then you write economic importance of bacteria in your classwork in the form of tabular column. Okay, thank you students.